Welcome back for more AP Biology. Today we're going to be moving on to our last topic of the year, which is wrapping everything together. We're going to do that by using a lab that we did in the past that confused everyone, and where we're going by revisiting the past. So we're going to try and figure out what's in common between everything, something that they test on. So where have we been? We started off the year by looking at the macroscopic view of the world. We realized that, we ran out of stuff to talk about. Can we zoom in any more? And the result is, absolutely we can, and that would be the microscopic world. But that wasn't all we can do. The question is, couldn't we zoom in just a little bit more? And the answer is, of course, and we zoom in even more and we get to a molecular world. The catch is, we could take all of these three segments of existence and we still have some questions because we still have yet to answer some basic questions from the start of the year, which is, where did all the diversity come from? Because we never really answered that question. We talked about evolution, but we never really talked about species and where species came from. So that's where we're going last. That is how we're ending the year. We started the year with the big picture, and we're going to end with the big picture. So if I look at all of life, what do we happen to have in common? Well, there's all sorts of traits that turn out to be in common between all living things. But we can do better than that, just outside of those traits. We can actually get specific, because we actually know specific things now. So if I were to take something really nitpicky, like ribosomal RNA, and start looking at patterns, what I can develop is a picture that looks like this, where I can actually see what organisms are most closely related to other organisms based upon the sequence of their ribosomal RNA. And the question should then become, well, if I can do this for ribosomal RNA, what else can I do this for? Well, if I look at just how we build organisms, look at how we build animals, it turns out that there are genes, transcription factors, that dictate what body parts get placed where. And those genes, those Hox genes, homeotic box genes, that dictate where body parts go, are interchangeable. Meaning, we haven't done this, but you could take a human and take the Hox gene that says, here's where your arms go, and you could steal one of the genes, a Hox gene from a fly, put it inside of a human, and the human will grow normal limbs. I could take a mouse gene that says, here's where the tail goes, put it inside of like a lizard, and it will grow its tail appropriately, and it won't look anything different. The parts are interchangeable. We have genes that seem to be conserved over evolutionary time. And if I look at those, like Hox genes with animals or ribosomal RNA with all of those organisms, we could make a list of things that are in common. This was part of your BLAST lab. You were asked to figure out what is in common. And a lot of people struggle with this, and the reason why you struggle is you're not thinking enough because you're trying to be too clever. You're trying to be too nitpicky. If I were making this list, I'd have things like DNA polymerase, RNA polymerase, ligase, helicase, primase, the enzymes that you find in glycolysis, so hexokinase and phosphoglucose, isomerase, the triose phosphate isomerase and aldolase and enolase. I'd be going after those enzymes because that is what I would find in common with virtually all organisms. I wouldn't be going grandiose. I'd be going with basic stuff. BLAST, when we use BLAST, what it does is it takes a sequence that you give it and it just says who matches. That's all it does. It just looks for matches. It's kind of like how we use Turnitin, teachers use Turnitin, and all it's doing is, where can I find the matches? So you could have a paper that's 50% plagiarized, but it's 51% pieces that add up to being 50%. I can do the same thing with a BLAST search, where I can find, how well does this gene or this sequence match other genes? That's all it does. It gives you no interpretation whatsoever. And if you're clever with what you hunt for, it can give you clever things in response. Among those clever things is it can build a picture that looks like this. It's referred to as a cladogram or phylogenetic tree. The way you interpret this is, if I were to find a point like this one right here that I'm circling around, all of these organisms down here turn out to have stuff in common. So whatever the trait is that we were looking at, whatever the kinase was, all of these are similar. So the crustaceans, bees, flies, grasshopper moths, horseshoe crabs, the kinetoplastids, and the kinetoplastids, so trypanosomes, which are protozoans that cause some weird African diseases. So if I look at just those organisms, they have something in common. And they would have stuff more in common than would like the birds and rodents way up at the top, because they're in a separate group. What I'm looking at are genetic ties. What is in common? And this came from one particular gene that was being hunted for. So could I do this for another gene? Sure. Could I do this for another gene? Sure. Could I do this for a different gene? Sure. I could do this for 1,000 different genes, and I could start to see how do these pieces start to stack and overlap. If I do that enough, I could build some patterns. I can build a history, in a sense, of relatedness. And if I do that across different species, I could start to ponder, how did these different species come about if they have things that are related to each other? That study, the study of how these things are, how we make all these different species and how they're related, falls under the category of what we call macroevolution. And the actual mechanisms of making new species is called speciation. And that turns out to be where we're going to go next.